With the COVID-19 pandemic troubling the world, a solution that many countries are betting on is a vaccine. However, with a lot of articles and videos on the side effects of vaccines, there is a lot of skepticism behind this drug and a lot of fear into its potential side effects. So today we'll go into how vaccines work, the different types of vaccines, and how they're made, so that you can make a better educated decision on whether you want to take this drug or not. So before we go into how they work, let's quickly discuss what the body's reaction is when exposed to an infection. We have the innate immune response and the adaptive immune response. The innate immune response is the body's first and fastest reaction. This response uses special cells in the body to detect anything foreign that has entered and to attack and destroy it. The purpose of the innate immune response is to attack anything that looks foreign in the body, whether it's bad, harmless or good, which is why you can get an allergic reaction to things such as nuts and pollen. This non-specific characteristic allows it to be fast and to destroy anything harmful that may have entered the body before it gets a chance to spread. However, sometimes this isn't enough and the pathogen manages to survive, spread and go into the body to cause harm. Such has been the case with many people who contracted the coronavirus. This is where the adaptive immune response comes into play. The adaptive immune response is more specific, unlike the innate response, and therefore takes a bit longer to work. They rely on antigens on pathogens. Antigens are distinct surface features on pathogens that are unique to each pathogen, and the adaptive response uses this to its advantage to recognize and mark the pathogen for destruction. They do this with something called B cells. These B cells are Y-shaped molecules where one end is known as the Fab region and the opposite end is known as the FC region. The Fab region is unique to the antigen on the pathogen, a bit like a lock and key. So when the Fab region recognizes the antigen, it binds to it. Then the FC region of the B cell calls for backup to destroy the pathogen. So in essence, the B cell is a tag that recognizes a harmful entity and calls for other cells in the body to kill that entity. One of the main benefits of the adaptive response is that once it has killed the harmful cell, it will form memory B cells that will remain in the body, so should the body encounter the same pathogen again, the B cell will instantly recognize and kill it. Therefore, the body is immune to it. The body can have up to 10 billion different B cells present at any one time, each with a unique Fab region to recognize different antigens on different pathogens, and therefore it takes time for the right B cells to come across the pathogen. And once it has recognized it, it has to produce multiple copies of itself so that it can go around the body and kill all the other pathogens with the same antigen. Unfortunately, in this time, the pathogen is attacking the body, and sometimes by the time the right B cells have been produced, it's too late. This is where vaccines come into the picture. The basic concept behind a vaccine is that it exposes the body to a weaker and harmless version of the antigen associated with a desired pathogen. This gives the body plenty of time to create the right antibodies to kill the pathogen, so that if the person gets infected with the real thing down the line, the immune system has the right tools to kill it instantly. So you get immunity without the risk of the pathogen harming the body. So what are the different vaccines? There are four main classes: live attenuated vaccines, killed or inactivated vaccines, inactivated toxin vaccines, and purified subunit vaccines. Live attenuated vaccines are, just as the name suggests, a live form of the pathogen, but a weakened form of it. This is done by culturing the pathogen in a really harsh environment so that it mutates into a weakened and non-pathogenic version. Yet it still maintains some of its structure so that B cells in the body can recognize it and develop immunity against it. Because these type of vaccines are so similar to the natural infection, they induce a strong and long-lasting immunity. Just a couple of doses can give a lifetime of immunity to the pathogen it's imitating. But it does have its disadvantages. As it is a mutated version of the pathogen, a lot of the antigens that it has on its surface may not be identical to the non-mutated version of the pathogen. This makes it difficult and expensive to develop, as you need to produce a mutated version that presents similar antigens whilst also proving that it is harmless and that it would never mutate back to being harmful. Examples of live attenuated vaccines are vaccines against measles, smallpox, and chickenpox. Killed or inactivated vaccines are, in the case of bacteria, a killed version of it, and in the case of viruses, an inactivated version of it. This can be made by killing the pathogen by heat, but this process alters the pathogen's shape, which can reduce or nullify the effectiveness of the vaccine. They can also be killed by chemicals such as formaldehyde, which has the benefit of not altering the pathogen's shape significantly. However, using chemicals such as formaldehyde can leave toxic residues, so it is difficult to achieve a concentration of chemicals that is strong enough to fully kill the pathogen, but at the same time weak enough to not cause any harm in the person taking the vaccine. The benefit of using this type of vaccine is that drugs for new strain of disease-inducing pathogens can be quickly produced. Moreover, production is easy and cheap, and safety testing is simpler. But as it's a dead version of the harmful pathogen, it can't go around the body and divide and therefore cause a strong immune response. As a result, several doses of the vaccine have to be given over time, and the immunity produced is relatively weak and short-lived. Examples of inactivated vaccines include hepatitis A, flu shots, and polio shots. The third class of vaccines are inactivated toxin vaccines, also known as Toxoid vaccines. One way a microorganism can cause harm to its host is by producing a toxin which then goes on to attack the body. So this vaccine involves extracting the toxin produced by the pathogen and inactivating it with chemicals such as formaldehyde to produce a harmless version known as a toxoid. So it's similar to inactivated vaccines but focuses on the toxin produced by the pathogen rather than the pathogen itself. The advantages of toxoid vaccines are that their purity is high and therefore it can create a strong immune response against the pathogen. Also, adverse reactions are rare. The disadvantage of this is that this form of vaccine only works in situations where the main symptoms caused by the pathogen are due to the toxin it produces, so it cannot be used for a lot of harmful microorganisms. An example of a toxoid vaccine is the tetanus vaccine. The last class of vaccines are purified subunit vaccines. This basically means they're made of specific sections of the pathogen. These specific sections could be part of the pathogen that play a key role in its disease-inducing ability, or they can contain the antigens that help identify that specific pathogen. The subunits can be extracted by breaking up the pathogen and then purifying it, or they can even be produced through genetic modification. And because the process of extracting the subunit involves breaking up the microbe, it automatically gets inactivated. The advantage of these vaccines is that they produce a highly specific and a strong immune response, providing strong immunity, and adverse reactions are rare. Example of subunit vaccines include the influenza vaccine. So that summarizes the different classes of vaccine. I hope you've enjoyed this video, and if you have, don't forget to like, share, and subscribe.